this is due just unfortunate flight schedule, but I try to be at least fast, so you can enjoy Professor Mog's presentation about the wonderful story of EGFR mutation. Actually, I have to confess that the story of EGFR mutation converted me also as a fan of targeted therapies, and the concept that if we can really find the real molecular cause of cancer, and we have a wonderful drug to target it, it really brings to a solution. I think it's a revolution what we experience now, very similar to what we experience in infectious diseases. When we finally found out why patients have pneumonia and identified the bacteria, the real cause, and we had targeted drugs against the cause of the disease. Now, we also know from the title of Professor Mog that it's a story of 10 years, and we all remember that in the first years after the discovery of EGFR mutations, there were many, many painful years while we were waiting to get access to EGFR TKI therapies, although we already knew about the significance of EGFR mutation. The question here, what are we going to do with the other 90% of lung cancer patients, and how can we accelerate the access of, to new targeted therapies? So actually, the change has already started. This year, the NCCN guideline already recommends not only the testing of EGFR and ALK, but also the multi-gene, next-generation based testing, which can be, these two genes can be tested in, as a part of a multi-gene test. And actually, especially in the US, the major cancer centers already started large cancer genome profiling services, like Dana-Farber Cancer Center now just upgraded its panel to Oncompass, which includes 305 genes. And these genes are tested in all solid tumor patients who come into. So this is part of now the diagnostic triage. You do a CT, PET CT. Of course, you have a molecular profile of the tumor, the whole, not just one, but the whole. You want to know what are the drivers, genes in the tumor. And also in Mayo Clinic, Washington University, all that have their own programs. And also we have private companies who provide service, not only the molecular profiling, but also the molecular interpretation. Now, the big question is whether are we ready to do this in Europe? Can we do this, and can we deliver the advantages of such approaches to patients in Europe, in our countries? So we started a program a few years ago. We started systematically to visit opinion leaders, oncology centers, especially those where many targeted clinical trials are ongoing. And we collected the information, what could be the therapeutically relevant targets? What should we test here in Europe for the maximum benefit of our patients? So we collected the 30 targeted drugs which are already in clinical use, and 200 in clinical trials, ongoing clinical trials. Although there are other 800 in preclinical research, but now let's of course focus those in which are at least in trials available. And we linked these drugs to 58 genes. And we created a panel for next generation sequencing of 58, and we added also the key FISH tests for translocations and amplifications. And nowadays, of course, we all know that molecular diagnosis is part of our lives, and we are getting used to it. But also what we have to learn in the future, that oncologists and doctors in general will have a new friend, a new tool, and this is informatics. And medical softwares will develop in the next years to help us to accelerate the decision making. Because when you have a report about 50 genes, 100 genes, with the potential of mutations out of 2.1 million reported in COSMIC, and you have to make a decision in 5 minutes what kind of therapy you choose your patient, that will be a big challenge in the everyday life. So this is why such informatic solution systems are being developed, we also use. Now, this is just some results in a small number of cases. It was we analyzed 86 non-small cell lung cancer cases where we sequenced these 58 genes and did the FISH analysis. Actually, we found that in majority of the cases, we found one driver. Now, this is the easiest thing when you have just one driver to figure out what kind of therapy to offer. But of course, we had many cases where not just one, but many drivers are present, and it will be a great next challenge in oncology, how to tackle cancers in multiple drivers. And also, there are some cases which were super wild type or non-driver mutation. Now, if you look at the distribution of these driver genes, what you can immediately see that there are some genes which are frequently mutated, but there are many genes which have been found to be mutated in a single patient, very rarely. Put it in another words, the frequently mutated genes are rare. This is a funny phrase, but it just suggests that the problem is that we are talking about now EGFR and ALK as a great example for targeted precision medicine, and we talk about the fact that they are rare, 5%, 10%, 1.7% like ROS1, but actually these are the frequent drivers. All the others will be less frequent, and it will be a great challenge how to match therapies and how to conduct clinical trials to prove efficacy of drugs against these rarer targets. Actually, based on this 58 gene test, we could find a positive association with drugs in 90% of the cases. Of course, in half of those were drugs in clinical trials and half in clinical use, and in 40% of the cases we found negative association, which is also a very useful information for clinical practice, whether you should give a drug or you should consider a patient to refer to a targeted clinical trial. Actually, in third or half of the cases, we identified driver genes which should be targeted, and in the other half of the cases, we actually recommended anti-EGFR treatment, although we only found, of course, the 5–10% mutations, and we also found some FISH positive, and we found in many cases non-driver super wild type patients, but we recommended EGFR inhibitors for those two. And the reason why is if you look at the distribution of driver genes in non-small cell lung cancer or adenocarcinoma, to be more specific, and you put the results of the erlotinib BR21 trial we all know very well, there is a very interesting overlap of the numbers. So you can see that in the trial, unselected adenocarcinoma patients were treated with erlotinib, EGFR inhibitor, and half of the patients progressed. And there was, of course, we all know that 10% subgroup who responded extremely well. Now we know that these are the EGFR mutants, but there was another group, 30–40% of patients who actually had a long, stable disease at least. So there is a group where EGFR is not a strong driver, but has a biological role in lung cancer. And I'm just wondering, this is just a hypothesis, and maybe the future will tell, that when we see those cakes, we always see the cakes of the different mutations in lung cancers, there is always a piece where it's said that no mutation found, unknown. And maybe this is not unknown, these are really wild type. So we will learn that maybe there is a fraction of lung cancers where there is no mutation-driven driver, and in these cases, the overactivation of EGFR is indeed is part of the game at least. Maybe because of the amplification, or what is even more likely is due to the autocrine upregulation of its ligand. But we noticed, we learned also in colon cancer, and there are some reports that it can be the case also in non-small cell lung cancer. But at least this is an example that we have to learn from the clinical experience and teach the biologists, not only vice versa. We have to see what our experience is in the patients. So what can we do if we find a driver mutation? Of course, NCCN now lists not only registered drugs, but also lists some indications when there is a drug which is not registered in non-small lung cancer, but it has a clear target. So this is just one example of our experience. This was actually one of our colleagues, unfortunately, he was an anesthesiologist, and he had a squamous cell carcinoma, and we found MET amplification. And although crizotinib is registered for ALK translocation, this patient will get a very special permission to have a crizotinib treatment anyway, and he responded very well, and we, of course, published this case. And also there's another nice story really close to my heart, because this lady actually even let her face to be shown and consented that, because she was living in a very small village, and she never traveled outside her country, never seen the sea. But we found an ALK translocation in her tumor 2 years ago when crizotinib was not available and definitely not reimbursed. And it's a big problem because we all know how much 1 month of crizotinib treatment costs. So, but luckily, we found a place for her in the seritinib trial in Malaga. And because of her husband was a truck driver, the company organized for her a free trip to Malaga. And local patient advocacy groups found a free accommodation. So, actually, this was the first time for her to see the sea and walk on the beach while treated with seritinib. And the very good news is that she's still in remission for 2 years now. And we also saw in the last presentations how well these ALK inhibitors work. So this is our overall experience, that how many patients get actually the molecular profile-based treatment at the end. In reality, and what we found is around 1/3 of the patients, so 20% of the patients actually were referred to targeted clinical trials, and 10% received on-target, target-based therapy based on the molecular profile. So we are happy that at least 1/3 of patients were treated based on the molecular profile, but of course we are not satisfied. This is extremely difficult to get access to these therapies, to get access to these clinical trials, and this is what we have to work much more and focus much more. So that's why we created a whole program around it, what we call Therapy Access Program. We collected a team of molecular biologists, genetic counselors, oncologists, and we tried to help each patient and each oncologist to really make this dream true, that the patient is treated based on the molecular profile. Of course, in reality, it's not just enough to know that there is a clinical trial in Malaga. You have to call the PI 10 times to make sure that the slot is ready. You have to communicate which additional tests have to be done before the patient goes, so it's not going in vain. But it's very difficult to squeeze this into the everyday life of an oncologist. So that's why we realized that this is the biggest hurdle now. We can sequence the whole genome, we can interpret the mutation. The biggest problem is how to get access to the drugs. So we will work harder and hope we can work together for many, many patients in the future. And there will be more and more winners of precision medicine. Thank you very much for your attention.